Hello and welcome to another episode of the Omics Cast. Today we're going to talk about 10 quick tips for effective dimensionality reduction. This episode is motivated by a tweet I saw on Twitter, which is pointing to this PLOS paper, 10 quick tips for effective dimensionality reduction, which is written by Lang Huang Nguyen and Susan Holmes, both at Stanford. And it's a PLOS biology paper. First author Lang Huang Nguyen, a PhD student in computational mathematics at Stanford University, and Susan Holmes, a professor of statistics at Stanford University. And so the paper is about how to use dimensionality reduction, which is frequently applied during the analysis of high-dimensional data. For most of things that happen in genomics or next-generation sequencing, omics in general, it can be beneficial for the majority of modern biological data sets. And it's not uncommon to have hundreds or even millions of some simultaneous measurements collected from a single sample. And then dimensionality reduction aims to solve one of the problems with statistics called the curse of dimensionality, where many statistical methods will lack power when applied to high-dimensional data. So even though we have methods to give us very high-precision readouts from biological data, the fact that they are very highly dimensional poses problems when we need to analyze those case controls or those samples with statistical tools. Even if the number of collected data points is large, they say that there's still good reasons to use dimensionality reduction. They remain sparsely submerged in a voluminous high-dimensional space that is practically impossible to explore exhaustively. And so they say low dimensionality data, low dimensionality representations, can remove noise and retain signal, and those will be available to further analysis of the data. And this is based on the premise that the measurements can be sometimes uninformative or redundant. And for a lot of what we do in genomics or omics, there are many data points that point into the same directional, and so that makes them redundant. And if you can reduce the dimensionality of what we do from readouts from genomics or any omics technologies, it reduces the number of dimensions that makes them more amenable for statistical analysis. The fact that a lot of these are redundant or non-informative means that dimensionality reduction, DR, can be viewed as a method for latent feature extraction. So you try to highlight what the latent features are in the data. And they talk about many DR techniques. They say that sometimes some are easy to misuse, and they result in often misinterpret in their nature, the way they are interpreted. And so they give 10 tips for applying effective dimensionality reduction. And tip number 1 is to choose the appropriate method, right? Some of what they say here is have a look around at what your field is using, and then try and see if you can describe your data in the different ways in which it can be represented once you apply the dimensionality reduction. They say there's two main methods, linear methods such as PCA, CA, MCA, or CMDS, or equivalence, or PCOA. And these are more adept at preserving the global structure, whereas nonlinear methods such as kernel PCA, NMDS, isomap, diffusion maps, or any of the varieties of the neighbor embedding techniques are better representing local interactions, right? And so this is a crucial aspect for it because sometimes in genomics or in omics, we look at the chromosomes in the human genome as the very long vectors from which we can take up measurements, and having a very good way of drawing where these interactions are global. For example, genes that express proteins or enzymes, and those enzymes inside the cell interact with each other. Those global interactions would be more amenable for things like PCA. But if there are local interactions, that are physical connections between one part of the chromosome and another, and it happens many times in functional genomics, then those would be amenable to another set of tools, right? They say if observations in your data have assigned class labels and your goal is to obtain a representation that best separates them into known categories, then supervised DR techniques are also worth looking at. And LDA is probably the most famous one here, but PLS and NCA and the most recent one, bottleneck neural network classifier, are also worth looking at. For situations in which multi-domain data are gathered, for example, gene expression together with proteomics and methylation data, typical multi-omics analysis, you might apply DR to each data table separately and then post-process with something like a Preprocessing transformation. That's one option. Or what you can also do is consider methods that allow the integration for multiple data sets, such as the conjoint analysis method for multiple tables known as statis, and the equivalent method for the conjoint analysis of multiple distance matrices, or DStatis. And so these techniques are more like pure multi-omics techniques that come from looking at this data more globally, and they may be more relevant for cases like this. So in this table of different methods, there's the ones that are more applied to global interactions, the set here for local interactions, and then the ones, as I said just in a moment, the supervised DR techniques here, and the bottom of the table. The last two statis and these statis are for multi-domain, or let's say multi-omics approaches. So tip number 2, pre-process continuous and count input data. This is something that a lot of people will be familiar with, which is to sort of do a little bit of data munging at the beginning where you get your table or vector of row count or row measurements to make them more amenable for the statistical analysis further down. So data centering is subtracting variables means from each observation, and it's applied quite as a standard implementation quite often in the preprocessing of data, and then scaling, multiplying each measurement of a variable by a scalar factor so that the resulting feature has a variance of 1 is also a very common technique. When the units of all variables are the same, for example, in high throughput assays, normalizing feature variances is not advised because it results in shrinkage of features containing strong signals and inflation of features with no signal. This can be the case where you are taking the measurements from a sampling process where you have a small long tail of measurements, like, for example, the rare genes that if you don't sample enough, the normalization may get rid of the signal that you have. Other data transformations may be required depending on the application, the type of import data, and the DR method used. For example, if changes in your data are multiplicative, for example, your variables measured percent increase or decrease, then you should consider a log transform before applying to PCA. And this is quite common, log transformation, because a lot of things are in that kind of scale in biology. A lot of things that are measured in molecular biology are that kind of scale. And then when working with genomic sequencing data, 2 issues need to be addressed before you can apply DR. First, each sequencing sample has a different library size, what's called diversity or sequencing depth, a nuisance parameter that artificially differentiates observations. If you've done 2 samples at different sequencing depths for something like, for example, digital gene expression, then you have to normalize for those, otherwise your data will look just off in general when you do a comparison across samples, across sequencing runs. So in order to make observations comparable to each other, samples need to be normalized by dividing each measurement by a corresponding sample size factor, estimated using specialized methods. And the 2 that they mention here are DSeq and EdgeR. DSIC and DSIC2, the implementation of DSIC2 of DSIC is probably the most common one, and HR is got also some use cases that are useful, and sometimes running both and comparing is probably the best approach between one and the other. The other aspect of dealing with genomics is that the data may exhibit a mean variance trend in which features with higher means have higher variances. This is also usually due to the sampling nature of some of these omics analysis. A variance stabilization transformation, or VST, is needed to adjust for this effect and to avoid bias towards the highly abundant features. For counts with negative binomial distribution, and these also happen quite often in biology, in omics, such as sequencing read counts, an inverse hyperbolic sign transformation or similar techniques are recommended. So sample normalization and sample stabilization together are very effective and sometimes efficient preprocessing steps for high throughput data, definitely for genomics and multiomics. Tip number 3 of the 10 tips of this PLOS paper: handle categorical input data appropriately. They discuss about some of the main differences between PCA-like methods and non-PCA-like methods, and then they say if the mixed data set contains a large number of categorical variables, and this can happen especially for the type of kind of molecular phenotyping data, MFA or multiple factor analysis can be used, and the method applies PCA on numerical and MCA on categorical variables and combines the results by weighing variable groups. Examples of these methods that we discussed before, this stat is the one that's a multiomics, is in R function. This stat is R. All the other ones, they have their own methodology in R almost equivalently. Some of them in Python, scikit-learn being here the most common, SciPy as being the underground library for many of them, but some of them don't. And this is one of the observations, I think, in the world of genomics or multiomics recently, which is Python and the libraries associated to Python, SciPy, NumPy, pandas, etc. All these are catching up to what people have been doing using the R language for quite a few decades now, 2 or 3 decades at least. But R is a still big and thriving community, and so some of these methods, they haven't caught up yet, because essentially to implement them in Python is to re-implement them in a similar way to where they were implemented originally, and then why not use them originally in R? So definitely Python is catching up, and a lot of these are going to be available for both, but there are some of these tools where the first go-to place is the R language and the associated CRAN libraries. So one method for categorical variables that is appropriate, converting categorical variables to dummy binary features, is one method. Another approach is to use optimal scaling categorical PCA or CAPTCHA. The optimal scaling places original levels of categorical variables with category quantifications such that the variance in the new variable is maximized. So we kind of stretch out this categorical data in a way that the quantities are stretched out for the further analysis. Okay, tip number 4, using embedding methods for reducing similarity and dissimilarity input data. I should say tip number 3 was about neighboring methods. Tip number 4, using embedding. So this one is using embedding methods for reducing similarity and dissimilarity input data. When neither quantitative nor qualitative features are available, the relationship between data points measured as dissimilarities or similarities can be the basis of the R performed as a low-dimensional embedding. They talk about some of these and how they can be turned one to the other. I wanted to point out here about a collection of neural network-based approaches called Ward-2-Beck and the equivalent gene-2-Beck for gene expression, which are quite common in genomics, in what they call co-occurrence data. And so these are useful tools that people are used to apply to methods, and sometimes by reading what other people have done in the literature, it is important to look at different common pitfalls and then apply to the data that you have at hand. Okay, let's go straight to tip number 5, consciously decide on the number of dimensions to retain. This is kind of a philosophical tip in my opinion, but in any case, it is important that you realize what do you want this DR reduced data set for? What do you want to apply after you've DR'd the data? It says, remember that the number of dimensions can be at most the minimum of the number of observations rows and the number of variables columns in your dataset. For example, if your dataset contains expression of 10,000 genes, but only for 10 samples, then the minimum of 2 is 10. So there could not be more than 10 axes in your reduced data representation. And so this is where your friendly statistician will complain to you about the number of samples, sample size, and the number of replicates, technical replicates, or biological replicates. And they'll say, for your case control analysis, for the things that you want to stretch out or differentiate here, unless you have more samples, you're going to be heavily biased towards an aggressive DR procedure here. And sometimes, trying to overanalyze data for which you don't have enough samples is just a waste of everybody's time. How do you know how many reduced dimensions to keep? Well, in practice, people usually rely on what they call the scree or scribe plots and the elbow rule when making decisions. A scree or scribe plot simply shows the eigenvalues corresponding to each axis in the output representation, or the proportion of the variance each axis in a PC, for example, explains. So this is a nicely looking scree plot that basically shows 1, 2, then 3 dropping quite substantially dimensions explain a large percentage of the variance, and then 4, 5, and 6 drop quite quickly. And then from 6 onwards, you can only explain a very small percentage of the variance. Tip number 6 of these 10 tips for how to apply dimensionality reduction effectively. Apply the correct aspect ratio to your visualizations. This is more for convenience of the visual checking of the results, but it is important to sort of understand what measurements you're dealing with, and if you plot them correctly, you can see trends, because the human brain is essentially, we are visual animals, and so the human brain is better at looking at things than machines, and to spot patterns and to make decisions based on the understanding of the data that we have. And so applying the correct aspect ratio helps in that. The proportional relationship between the height and the width, and also the depth of a 2D or 3D plot, can strongly influence your perception of the data when you're doing visual validation. DR plots should be consistent with the relative amount of information explained by the output axis displayed. A way of doing this is to use ggplot2 in the R package for generating plots and adding the coordinate fix1 flag, which will ensure the correct aspect ratio. So this is one way of looking at it, in a couple of sort of dummy examples of rectangular, square, adjusted racial label, and adjusted racial label plots. Tip number 7, understand the meaning of the new dimensions. And here we're they show an example in the paper. We're gonna concentrate on the fact that original variables importances to the new dimensions can be visualized using contribution bar plots. And then sometimes this is a very good way to immediately perceive what are the relationship between these factors that you're looking at in your tabular data. When working with high dimensional data sets, such as high throughput assays, you can limit the plot showing only the top few, for example, 20 features with highest variance or highest variability that you have in your contribution in your data. And they show an example here for, I think this is a wine example, wine data set showing how variables representation can be used to understand the meaning of the new dimensions, right? Variables and observations can be included in the same graphic, referred to as the biplot. Biplots, such as the one in figure 4, allow you to explore the trends in the data samples and features simultaneously. We don't show this, but it's quite similar to this plot here. Badge effects, another important aspect of dimensionality reduction and data analysis. Badge effects are technical or systemic sources of variation that obscure the main signal of interest. And you have to deal with them because otherwise they'll cause problems downstream. If a batch effect is discovered, you can remove it by shifting all the observations in such a way that each batch has its centroid, the group's very center, located at the center of the plot. So it's a way of sort of in the optics world focus in so that all the points that you want to look at are in focus, are aligned. Tip number 9 and penultimate, take advantage of multi-domain data. This is again in relation to this multi-omics analysis that people are performing these days, sometimes at the single sample level, single cell level, where they can say, let's look at both digital gene expression, microRNAs, methylation, ataxic, etc., and you have all these different measurements for almost the same data points, meaning single cells or something else. One way of doing this is correct for every one of them and then apply some sort of adapted PCA, but there are methods nowadays that can be applied directly. For example, statis and distatis. Both methods are used to analyze several sets of data tables collected on the same set of observations, and both are based on the idea that combining data sets into a common consensus structure that is called the compromise of what explains the most, giving all the multi-omics applied. And finally, tip number 10, check the robustness of your results and quantify uncertainties. Essentially, for some data sets, the PCA PCs are sometimes ill-defined, and 2 or more successive PCs may have very similar variances. You can spot this in the scree plot, and it's a good way of knowing if the PC has been applied to the satisfaction of what's needed. When some of the scree plots flatten very quickly, you can say that these PCs are unstable and the dimensions corresponding to similar eigenvalues need to be kept together and not individually interpreted. Sometimes people have the tendency to overinterpret PCs and say, oh, if PC1 is this and PC2 is that, then PC3 must be something else that I think is a good candidate to being. Sometimes this is not relevant. It doesn't work that way. And in regards to methods that need parameter setting, the stability of the results against different parameters is important, and you can avoid issues like this by doing parameter sweeps. An example of this is the Tisney method, where you need to pick a value for perplexity. There is a method proposed by Cao and Wang to be able to use a big type rule for selecting the perplexities of a Tisney. Although in practice, your data is going to be somewhere in between the parameter suite that you can run anyway. And these, many of these methods are not computationally super expensive. So in practice, you should repeat your DR computations for a range of input parameters, say for Tisney, the perplexity value. Finally, a separate concern is the method stability against outliers. And outliers are quite common in biology and microbiology, multiomics, but difficult to describe. I mean, what you think is an outlier, someone else may think it's giving relevant information to the study. In the paper, they say, in the case of PCA and other linear methods, if all of the points in the sample projection plot are located close to the origin, and then there's only a few points that lie very far away, the DR solution is said to be dominated by the outliers. Right? Here is the scree plot of a case where the values are very close to each other. Right? So there isn't a PC 2, 3, and 4 really, or a meaningful one, because as you can see from here, they explain quite a lot of the same. Right? So coming back to outliers, if samples are removed, the DR needs to be recomputed and the changes in the output representation should be noted. Additionally, you can estimate the uncertainties associated with observations by constructing, this is another aspect of it. how can you check how stable your data is? One is to construct a collection of bootstrap data sets, random data sets of the data generated by resampling observations with replacement. And then the second other way of doing it is when realistic noise models for the data is available, instead of using bootstrap samples, you can generate copies of all the data points by perturbing the measurement values, right? And they mentioned a case where statis and this statis will allow you to do that for the coordinates of the compromise. And that's it. Those are the 10 tips for effective dimensionality reduction. As a conclusion to this paper, they say DR is very useful and sometimes essential when analyzing high-dimensional data, and many of the multi-omics data is of that nature. One aspect that is useful for this, especially if you're working within a team of more than one data scientist, is to keep track of all the decisions you make, including the methods you select, the distances or kernels you use, and the values of parameters you use. And I would add here the different methods that you have tried, even if they haven't worked out very well. The most convenient way to save all the steps of your work together with the results obtained is through something like a notebook, like an R on an IPython or a Jupyter notebook. Once you can keep those, you yourself retrace the steps that you made to reach a conclusion, a result in your analysis, but also check with someone else that can validate what you've done or help you test some other aspects of the work. And that's it for this edition of the podcast. I hope you enjoyed it. Follow me on Twitter if you want to learn more about this, twitter.com/Alberto. Contact me on LinkedIn if you have any questions. I'll try to answer. tinyurl.com/Abiledia LinkedIn. And as a reminder, this slide deck is available upon request. Until the next episode, goodbye.